Have you ever wondered what drives our motivation to exercise? The pursuit of physical activity is often fueled by a profound sense of pleasure, a rewarding experience that keeps us coming back for more. But what exactly compels us to embrace exercise? To answer this question, we will explore the insights from a groundbreaking research paper titled "A Microbiome-Dependent Gut-Brain Pathway Regulates Motivation for Exercise." In their investigation on mice, researchers stumbled across a remarkable connection, a direct link between the gut and the brain. This connection holds the key to enhancing exercise performance by boosting dopamine signaling during physical activity. What's even more intriguing is that this connection is orchestrated by their microbiome, those tiny organisms residing within the digestive system. These microorganisms play a pivotal role in producing endocannabinoid metabolites within the gut, which in turn kickstart a chain of events that boost the signals of dopamine in a specific region of the brain called the ventral striatum. Stimulation of this pathway was found to improve running performance, whereas a disruption of this pathway was found to act as barriers that limited exercise capacity. In the following sections, our group will delve into the design and results of this study. First up, Evelyn will cover the metagenomic impact on exercise performance.
The first chapter deals with the metagenomic effects on exercise performance. 199 mice from 8 genetic backgrounds were studied and profiled for their genome, metabolic analysis, serometabolomics, and microbiome. The untrained mice then underwent exercise profiling by running either in wheels or on a treadmill. Data seen in both figures revealed huge variability between the mice exercise performance. So what causes this variability? Could it be the host genome? Probably not, as genome-wide association studies and genotyping have shown. Turning to non-genetic parameters, the researchers had to use a variety of tools to cope with the large variation across the animal cohort to show that the metabolic and behavioral traits did not correlate with the exercise performance. So what next? Could the regulator of exercise performance be our microbiome? To find out, a loss of function and gain of function experiment was performed by using broad-spectrum antibiotics or transplanting microbiota, letting the mice run again after interventions, and using isogenic mice to identify the taxonomic elements of the microbiome that determine its contribution to physical performance. Ablation of the microbiome was shown to reduce endurance by about 50% compared to control mice. Finally, to narrow down the possible candidates of the microbiome involved in this phenotype, the researchers reduced the range of antibiotic combinations administered, which also led to reduced running performance, as figures are showing. These results highlight that certain taxonomic elements of the microbiome are necessary for exercise regulation. But which ones? After more tests, initial results predicted several members of 2 families as possible drivers for exercise performance. Therefore, germ-free mice were colonized with those members of either Erysipelotichacea or Lachnospiraceae, or R. gnathus. Successful monocolonization led to the final results shown in the graph. Eubacterium and Copracoccus enhanced exercise performance compared to germ-free mice. To summarize the first chapter, these data demonstrate the role of the microbiome in regulating exercise performance and highlight certain members of 2 families as possible taxonomic contributors.
Thank you, Evelyn. Now let's discuss the impact of the microbiome on striatal dopamine. The effects of microbiome on striatal response to exercise were studied by sequencing the mice striatum before and after exercise, and medium spiny neurons expressing dopamine receptors 1 and 2 were identified, and they examined the expression levels of ARC and FOS, which are neuronal activity markers for dopamine receptors. Interestingly, the levels of ARC and FOS were significantly decreased in antibiotic-treated mice after exercise, and this suggests that the colonization of the gut microbiome is crucial in exercise-induced activation of the striatum. They then assessed the striatal dopamine levels of antibiotic-treated mice by measuring dopamine levels in both groups before and after exercise, and dopamine levels in antibiotic-treated mice significantly decreased after exercise, and this clearly demonstrates the importance of the microbiome for striatal dopamine response to exercise. Next, they examined whether exercise-induced dopamine response was mechanistically involved in the performance-stimulating effect of the microbiome, and they found that the distance covered by outbred mice increased with elevated dopamine levels, and the germ-free mice with microbiome transplantation from high-performance outbred mice or conventionally colonized C57BL6 mice showed increased dopamine levels and enhanced exercise performance. Control group exhibited decreased exercise performance when dopamine signaling was blocked with sapiroid, or the activation of dopamine signaling with bromocriptine restored exercise performance in the antibiotic-treated group, and this indicates that striatal dopamine as an important mediator of microbiome impact on exercise. Next, they examined how microbiome ablation impairs exercise-induced striatal dopamine levels. They first measured the levels of MAO in both groups before and after exercise, and interestingly, MAO expression levels were not suppressed in antibiotic-treated group. Next, they administered paraglin into both groups and noticed that the antibiotic-treated mice had increased dopamine levels and restored running performance, and this showed that the colonization of gut microbes enhances exercise-induced dopamine response via suppression of MAO activities. Now I will pass on the time to Christabel.
Now I'll be talking about the role of sensory neurons in exercise regulation. An experiment with innovated TRPV1 mice was done and showed that there was an impairment in their exercise capabilities.
which was similar to microbiome-depleted mice. Capsaicin, a chemical found in chili, was able to restore the exercise capabilities in antibiotic-treated mice. Since TRPV1 is largely expressed by sensory neurons, there was a need to determine if the spinal or vagal efferent neurons were the primary mediator of the microbiome impact on exercise. This was done through examining the dorsal root ganglia and nodose ganglia in exercising mice. The results showed that exercise-induced neuronal activation in antibiotic treatment was reduced in DRG but not in NG, suggesting the sensitivity of the spinal pathway to microbiome-derived signals. Similarly, the removal of vagal efferents did not affect running capacity of the mice, but the removal of spinal efferents did so. With regards to how sensory neurons affect mild dopamine pathway, the removal of spinal efferents lowered dopamine levels and elevated MAO-A transcripts, which is not ideal as previously discussed. To determine whether capsaicin's exercising stimulating effect is dependent on dopamine signaling, control and microbiome-depleted mice were co-treated with capsaicin and sulpiride. Sulpiride, being a dopamine antagonist, blocked exercising-enhancing effect of capsaicin in both microbiome-depleted and control mice, implying the need for dopamine signaling. Collectively, these findings demonstrate the impact of the microbiome on dopamine levels in the striatum and exercise performance requiring TRPV1 positive neurons. Now, I'll pass on the time to Esther.
Thank you, Christabel. Next, let's look at how exercise performance is affected by the gut microbiome. To determine what exactly stimulates DRG neurons, the researchers used differential antibiotic treatment and untargeted metabolomics. Numerous fatty acid amides, or FAAs for short, were among the most potent metabolites. OEA is one example. OEA levels were lowered by antibiotics that impaired exercise performance, suggesting that FAA metabolites may stimulate sensory neurons to promote exercise. When an FAA diet was fed to antibiotic-treated mice, it recovered DRG activity, striatal MAO-A expression, dopamine levels, and exercise performance, all in the absence of the microbiome. Furthermore, gastric infusion of FAAs increased dopamine release induced by physical activity, suggesting that intestinal FAAs increased the motivation for exercise. It was also found that certain commensal microorganisms contain a biosynthetic gene cluster responsible for FAA biosynthesis. The essential genes of the cluster were isolated and introduced into germ-free mice. The results showed enhanced running performance, which highlights that FAA production by the microbiota might enhance host exercise capacity. Several FAA metabolites are agonists of the endocannabinoid receptor CB1, which is expressed by TRPV1 positive DRG neurons. These findings suggest that CB1 stimulation by FAAs may drive sensory neuron activity, striatal dopamine release, and exercise performance. To conclude these results, gut metabolites produced by the microbiome can elevate exercise performance and increase motivation to exercise.
Thank you, Esther. Put together, the paper highlights how the gut microbiome produces FAA metabolites, which trigger CB1 receptors on the TRPV1 sensory neurons. These neurons then send signals to the striatum to suppress dopamine degradation. Ultimately, higher levels of dopamine increases motivation for exercise. So, what does all this data mean? Firstly, this emphasizes the importance of the gut microbiota as a mediator in the mechanism underlying the runner's high, where prolonged physical activity itself generates feelings of reward and pain suppression to sustain exercise. From an evolutionary standpoint, this allows the gut to regulate one's capacity for exercise. Moreover, this research also highlights how this pathway is not linear. Exercise actually provides a reinforcing effect on enhancing communication in the gut-brain axis via the TRPV1 neurons. Now, this brings us to possible future research opportunities. The CB1 receptors in this pathway are known to be highly involved in mammalian energy regulation, from lipogenesis to satiety hormone regulation. Hence, the gut microbiota can possibly influence exercise performance in other physiological ways. The author also raises the term interoceptomimetics, which describes molecules that bypass the central nervous system and affect afferent neurons to regulate brain activities such as those involved in motivation. Hence, interoceptive interventions like metabolite supplementations and diets could present simple yet highly targeted and non-invasive methods to improve exercise motivation. However, these results cannot yet be generalized to humans, as current literature shows significant differences in the gut anatomy, genetics, and physiology between humans and mice.
In conclusion, the insights we've gained today open an exciting new chapter in our understanding of exercise motivation. It's becoming increasingly evident that our gut microbiome holds the keys to our exercise drive. And this revelation paves the way for the development of gut microbiome-related methods to boost exercise motivation and improve public health.